And now some words about data hygiene, open science, and open data. First, a word, just that these lectures are so far have been very focused on projects in science. And they're targeted for students who are leaders. If you're a leader of a project like it's an honor thesis, and actually a lot of this content is very graduate student centric. I would just say this class in general, especially if you're in the grad student section, you've got this other big project that the undergrads aren't doing. But the content and sort of the structure of it is really oriented around graduate students. So you should feel good if you're a grad student in the class that you're getting directed content, because this is, especially what you're about to see, is really focused. In fact, the last couple of lectures are really focused on trying to empower you. This is the empowerment project. To have a successful program, regardless of who your mentor is or where you are. If you're at, you leave here and you go to the University of Tennessee or whatever, this stuff applies. And what I'm about to say also applies very strongly across, no matter where you are or what you're doing. Okay, there we go. So 3 different areas of focus here: data collection and protection, data products, and data analysis best practices. My guess is this is going to be completely new content for you. So buckle up, maybe watch this more than once, and dig in, because this is definitely a way for you to become more powerful. Okay, data collection and protection. If you're leading a project, you must keep a laboratory notebook or a field notebook in which you outline your observations and activities. I would suggest something that has a tough cover and waterproof pages. Now, some people are really quick at texting, and you might text into your phone and just have like, oh, I'm going to have notes and put them in my phone. But then if your phone goes in the toilet, then what happens? Which happens to people sometimes. So I think you should ponder this very carefully. I am not opposed to innovations and digitization. However, generally speaking, old school lab notebook is still a crucial tool. And you should get into the habit of writing stuff in there every day if you're working in the lab. And I understand that you might be like, I'm doing the same thing every day. Why should I have to write something down? It's just a good habit. It's a good practice. And you may make observations that later become very important, especially if you're doing field work. If you're an ecology type, you might make observations in the field. But even if you're in the lab, you might make some observations about practice or things that are happening that become crucial later. So this is a really important thing for you to start as a habit and just never break. Keep doing it. Data should be collected. The notes should be clean and legible to others, written in pencil on waterproof paper with dates and data collector information there. So we know who collected the data, how they collected them, and what's going on. I believe this is superior to digital methods. I understand that some things, like some balances, for example, will put data straight into a computer or whatever. I mean, I'm not saying don't ever do that. I'm just saying, generally speaking, having written field notes or data sheets or lab notebook is very important. And I say pencil here because you can, if you're using ink, the ink can get ruined by water. So you can use Sharpie or other permanent ink. If you're sure it's permanent ink, that's okay. Like a fine tip Sharpie is okay with me, but I prefer pencil for all lab activities. I haven't gotten to the point, maybe I did this once, literally took a pen out for someone's hand and threw it out the window as a joke. But that's kind of how I feel about it. And here's a true story time. True story. During my postdoc, I hired a student, an undergraduate student at the University of Kentucky, to work in a greenhouse where I was doing an experiment. And I had already given her this whole speech about pencil, but she didn't listen to me, and maybe just not in a mean way, but just like, whatever. I say many words, and so maybe those words didn't catch. Anyway, so she was collecting data. She had to come in every single day to the greenhouse and record information. Like, I don't remember what we were recording about. And it was a pen. She was collecting in pen. And there was a clipboard with the data sheets on it. And we had a hellacious storm one day. And I don't know how this happened, but for whatever reason, the greenhouse leaked, which is not a thing that would happen. Greenhouses don't leak. It's like your house leaking. They're not set up to leak at all if it's a good facility. Anyway, it leaked directly on top of that clipboard. And the clipboard got soaked and all the data ran. The ink was gone. You literally picked it up and looked and there was nothing there. So it was a summer's worth of data lost. And true story. What I had the student do, she had to start spending half of her day in the greenhouse and then half of her day under looking in a microscope. Because if you look at a really powerful microscope at a sheet where you've written with pen, the actual ballpoint of the pen presses into the sheet. You can literally see the pressing. So like a pen like this, if I were to write something on this against a solid background, there it presses. And so she had to go reconstruct the data from the pressings. So that's how she spent her time. That wasn't me being mean. That was just like, that's the situation we're in. The data have been lost. So don't write in pen. Here's an experiment I did because when I told students this in 2020 or some other time I taught the class, some people were frowning in the audience, that is to say in the classroom. And so I thought maybe they don't believe me. So I went home and I took a sheet of paper and I wrote in green pen, I wrote the word pen. In blue sharp tip Sharpie, I wrote the word Sharpie. And in pencil, I wrote the word pencil. And then I put that underwater for like 5 seconds. This is what we got. See this? Gone. Don't be that person who's lost all of your thesis data because you wrote in pen. Pro tip, hashtag. Does hashtag go before or after? I can't remember. Someone has to tell me. Social media. Okay. Data collection and protection, item C. When possible, there should be uniformity in data collection system. This is a way to reduce error. Remember, I was talking about error before. There's all sorts of sources of error. One thing you can end up with if you have different people recording information is personal bias influences what you're recording. Now, and I don't mean that in like, it just is a factual reality. People are biased. So the way you record something might be slightly different. And if you have some kind of pattern in who's coming into the lab or whatever, you could literally sort of plow into your whole research this bias in such a way that it makes an interesting result. But it's got nothing to do with the flies or the whatever you're working with. It's got everything to do with who happened to be sitting there, like this person who's a wonderful former student in my lab, who's an ophthalmologist now, by the way. Anyway, so you should have the same person collected the same data in the same way for the whole project and only deviate if absolutely needed and with caution. And as an undergraduate, especially if you're just getting started in science, somebody's going to tell you what to do probably when you go in the lab. And at times you might think this is very, some of those instructions are very niggling. I had a doctoral student, story time number 2. She keeps track of how many stories. I had a doctoral student years ago who was extremely rigid about the details of everything that happened in the lab. And it was her, what she was doing is trying to eliminate interpersonal variance. Like if Susan comes in one day and Jim comes in the next, and you're working on the same balance with the same leaves, we can't have Susan and Jim do anything differently. They have to do exactly the same thing in exactly the same way. We can't have any variance. Better to have the very same person do it, but if you have multiple people coming in, you better be damn careful because you could have a big problem. And it could be something you can't sort out later, that you're like, oh, I've got this pattern in the data. The pattern's related to whether Amy was there or somebody else. That's unsolvable. So be very careful about this. Assess your data for quality and errors. So errors are quite common in data collection and data entry. You should practice quality assurance and quality control regularly. It's not the most exciting part of science, but if you don't do this, again, later you could have mistakes in the data that become difficult to deal with. Or maybe you don't even catch them and they end up in your final results. Now you've got fake numbers because somebody made a mistake. So a simple thing to do, again, if you're in the lab, in your lab notebook, however you're collecting the data, or if you're in the field and you're collecting field data like these two individuals, is make a quick scan of the data sheet to see if anything stupid is written down. If you have a tree seedling that has negative height, okay, it's not a possible thing. Or if you've got like blood alcohol or like listeria count, the listeria count is negative. That's not a thing. Can't go to zero. They can't go below zero. There's no listeria. That's it. It can't be negative 1,000 listeria or whatever. It's like you can't have absurd numbers. Also, during data entry is a good time to check for mistakes. And what I implemented years ago, and we're not doing this in my lab now, but probably should return to this, is when it's time for data analysis and you're all done with the data collection, get the sheets back out and literally check 10% of them and make sure that 10% of the things, 10% of the sheets, I mean, and that the data are perfectly entered. If you could, that's like, a month later or something, you haven't looked at them for a while. And if you because that like will separate you from the process and make you think about it differently. And if you find any mistakes, then do another 10%. Just keep going until you're like, the data are clean, crisp. Oops. Take precautions against the loss of data. This is a thing probably now in the era of Google Drive. It's less important, but still something to think about. It's just that computers are vaguely reliable. We have to assume they're going to fail. Something's going to happen. There's going to come that moment where it's fried and you don't know what happened. Somebody spilled their coffee into the keyboard, and you can't have vulnerability to your thesis project based on whether your computer works or not. What if you drop it down the stairs or whatever? It's like this stuff happens because we're fallible and computers can break. So you have to take precautions against the loss of data. Here's a side point, and this is where we're really suffering a little bit from not being in class together. This would be a point that might generate some conversation. It's important for you, if you're a student, which you are if you're in my class, to understand that the University of Dayton is the ultimate owner of the data you collect as a student or employee. They technically are responsible. The university as a whole is responsible for the data, especially if it's on a funded project. The principal investigator, your mentor, or whoever you're working for directly is kind of the proximate owner. This is my job in my lab. I am the person who's responsible for maintaining the data that the University of Dayton ultimately owns, if that makes sense. And then if we've got an agency involved, the agency also owns the data. If you're funded by the National Science Foundation, National Institutes of Health, or some other agency or organization, they own the data. So, and the responsibility flows like this: agency, university, PI, student, or technician. What can happen is if you're a student, a grad student or an undergraduate student, you're working on a project and you're putting blood, sweat, and tears into the collection of these data. You're going into the lab at night to test check the assays, the fish, are they still alive or whatever, seedlings in the field or whatever's happening. You can start to feel like this is my project, y'all. And for real, it is your project. That is true. You have earned ownership of that project. But it does not mean that you own the data. In this way, for example, if you're a doctoral student at UD and you collect the data and then you're just like, oh, you know what? I don't like being a student anymore. I'm going to take a job as a ski instructor in Boulder, Colorado, and go start teaching people to ski and screw this academia. See you later, y'all. Does that mean that all the work that went into collecting those data disappear with you onto the slopes of Colorado? No, it does not. That would be a disaster. It would be a disaster for the university and a disaster for the agency. But we have to assume that that's kind of what's going to happen. So what's the reality is you collect the data, you own them in terms of like being an author on the publication, being the author on the data set and so forth. But ultimately, it's the responsibility of the PI and the university, and ultimately the agency should enforce that to make sure that these data don't get lost. And it's not just like some fluke of the particular individual who's collecting them. In fact, agencies like the National Science Foundation literally require you to ensure that the data, material collected, archived, digitized, and made available allow current and future investigators to access the data and material. So literally, if you're funded by NSF, which I am right now, and others have been in our department, et cetera, you might work on an NSF project, you literally are required to share the data. The idea of going off to do something and just, I'm taking the data with me because I collected them all is not a thing. It's actually not even legally possible. I mean, it would happen, but this is like part of the agreement for getting the money is that you're going to share the data. So what do you do to make sure the data aren't lost? Well, this is what I recommend. Every project in a lab, if you become a researcher somewhere, if you're a researcher right now, you should have a shared drive because we're in the era of cloud computing and shared drive. And so you should have a shared drive with your PI, maybe with your committee, with your collaborators. And in that drive, you can have folders. The folders are literature, analysis, writing, scripts, data. And then you just put everything in there and work in that folder. I'm hoping that cloud-based system would be safe against data loss. So if your computer breaks, it doesn't really even matter. Just log on with another computer. Your data should be safe in that fashion. It would take something really bad happening, like an asteroid. Then at least you've got the data in the data sheets. If we have an asteroid that destroys Google headquarters. Okay, so here are the points about data collection and protection. We've summarized. If you're leading a project, keep a field notebook or a laboratory notebook. Collect data in a way that's clean and legible to others and in pencil. Let me also point out, I have had times in my career where I was looking at data sheets and entering numbers from the 1980s. So you need to write the data into your data sheet as if somebody you've never met before is going to look at them 20 years from now. That's what I mean by legible. Okay, you can't just make notes to self. This is notes to the world, to posterity. When possible, there should be uniformity in your data collection system, so you don't have random people collecting the data. Assess your data for quality and be responsible and take precautions, including creating that shared drive, as I mentioned before, which we'll circle back to this. Data products, item 2. Okay, so here first, I want to introduce you to the overarching philosophy of open data. And before I say that, before I go to the next slide, I want to say these words out loud. And again, it's sad that we're not together, so we can have some shared energy on this. We live in an era where there's a lot of skepticism about science. There always has been, but it's been amplified and brought to the forefront by charlatans and media organizations and politicians who are, in some cases, simply seeking power through dividing segments of society against one another. And they don't care why. They don't care. The only question is, do I get more money and power from this action, regardless of what's real? And that's just an awful reality of the society we live in. It's always been true, but it's worse now than ever before. There's lots of implications. We could have a whole class about this, like what to do, social media era or whatever. But what you need to do, my requirement for you, student in my class, is for you to take your own responsibility and take the act of being a scientist seriously. That's part of what the class is about so far. Take this seriously. Even if it's a small project, you take it seriously. You treat the collection of data with some reverence. And part of it, if you want to help make society better, is committing to openness about the analysis you're doing, the data you're collecting, making sure the data don't get lost, and having a commitment to sharing your data. An open data commitment. I'm going to collect these data and I'm going to share them, and then I'm going to do an analysis. You have to commit to that as a kind of your personal responsibility and personal ethics. And I think it'll help make the world a better place. The more people can trust the information, the better. And part of my philosophy for the class, as I've already mentioned, is I want to teach you to use R and also show you where a bunch of data are on the internet, et cetera, and just get you into the habit of like, I can go get the data and just analyze it myself. As long as people aren't lying about the data that got collected. And I've already talked about that too. So, hopefully you don't have to let somebody else filter the information. You can go get it yourself and download it and then do the analysis. So you also want people to be able to do that with your data. So this is a traditional scientific workflow, and this comes from this guy, Chris Lorty. And you can check him out. He's a very, very interesting guy. These figures are from him, from a talk I saw him give a while back. Collect the data, analyze the data, write up a paper, publish the paper. This is how science worked for however many years, up until 10 years ago, let's say. A lot of people, including this guy, Chris Lorty, don't like this model. It's not transparent, so the published paper is what people see, okay? The problem with this is I never see this model, the data, or this analysis. I see the figures and results, but I don't see the analysis. So it's not transparent. Also, if you collect data that you never write up and publish, what happens? That's scientific money spent that's down the toilet. And maybe crucial information is there that someone else would analyze and go, oh, here's the answer to this really important question, but I never got to see it. The person who did the thesis didn't publish it. They went skiing, or they went to Sarasota. The classic grad student finishes their thesis and like gets a job in Sarasota. And just like, this is friggin' awesome, man. I'm going to the bonfire tonight. And the PI is back oh, here we go. I got it in the next slide. The PI is like, hey, where are the data? Student graduates, PI is too busy to pay attention. Student gets a job, 13 months pass. The PI is like, where are the data? We need them as a baseline for our new study. The student, via email from Sarasota, Florida, does not reply for 7 weeks. And then writes, the data are on the laptop in the lab, but the laptop is gone. Got lost. Somebody dropped it down the stairs. This is not good. This is like the thing that's happened in science over and over and over again for 50 years. The students go to Sarasota, proverbially, maybe it's not Sarasota, maybe it's here in Dayton, but they disappear. They finish their thesis. While they're working on their thesis, the data is like, this is the thing. Once they defend, they're like, not in a bad way. I'm not mad at anybody who goes to Sarasota. That sounds good. But this is no way to do science. This is a problem because the data might actually be very important, right? But if you're in Sarasota and your PI is like, this fly eye data, or we have to get this out, and you're there and it's been 3 years since you worked on the project, you're just like, fly eyes are not important. Going to the bonfire tonight is, and I'm hoping the person at the bookstore will go with me and we'll have a hot date on the beach. That's important. Fly eyes do not matter to me at this point. In the nicest way. You like your PI, hopefully, but still. All right, so what are we going to do about this? Chris Lorty's got a plan. And this is just one example, but this is a very important example because many of you might become grad students and eventually finish your thesis and go to Sarasota. Here's a better workflow that addresses that issue. Collect the data, analyze, publish the data set. Aha. Ding, 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 ding. Now you can go to the bonfire. No one cares. All right. I've got the data. So does everyone else. Publish a data descriptor. This can go with the data set. So now we know what, not just the data set, but what's happening there. What do the variables mean? It's not good to have a data set that has some letters at the top of columns and not know what those are. FB, what is that? Or have like species names in a data sheet. It's just like some initials somebody made up. TM, what's this? Don't know what that is. Nobody knows. So that's the data descriptor. Then when this is all done, then write your paper, then publish your paper. Great news. You're not hindered in any way from publishing. And if you don't publish and you go to Sarasota instead, head to the bonfire, I'm still okay. And the field is still okay because we have the data already. This is the point. You can get this point through your head, you will be much more powerful and you'll make science better. Go. So this is why I want you to organize your project around the creation of data products. This is something completely different. Your PI probably has no idea what this is, okay? It's quite possible. They don't know what's going on here. So I'm not asking you to ask them. I'm asking you to do it. You just do this, okay? It doesn't hinder anything. You do it. Data products. What is a data product? Number one, a final CSV file that are perfectly clean and ready to be read into R. So we're going to work on this. We're going to work with some data sets that are poorly arranged and get you into the habit, and I'll teach you some skills. And like, you'll be able to work with a spreadsheet program to get the data arranged and ready to go into R. Also, already mentioned this, quality control, quality assurance, and EDA. Remember we talked about this the last lecture. Exploratory data analysis, doves, and that sort of thing, so that you know, like, here's an outlier that's stupid. A tree that has negative height. Trees can't have negative height. They can only grow up. Or here's a diameter of this fly's wing after we've mutated it. The length of the wing is 1.5 miles. It's not going to work. It's not one of those. It's not going to be the case. It's just put the data in wrong. So that you don't have any of those nonsense in the data set. So then you've got a final CSV file, perfectly clean and ready to be read into R. Step one. This, P.S. Don't ask me about this. You do this. This is what you do. You already know. Well, look at that. That's clear. You know what to do. Step two. You need metadata that describe all of the pertinent variables in the data set. So oftentimes what you'll have is a tab in an Excel sheet, okay? You can do this with Google Sheets too, or some other sheets. Is there like Apple Sheets? I don't know. Whatever, it doesn't matter. A tab in Excel that says, here are here are the details of all the data that are collected here. Do you need units? Probably, right? Like if it's, whatever, meter squared or something, or centimeters, or what, if that information is there. Are there any like initials or whatever, the JB? Okay, what is that? Like, remove all ambiguity around what's in the data set in a tab. Just type it out. Here's what every one of the column headings means. And you can put notes in there too. Here's what happened. My old, this wonderful person who was a lab manager at a place where I worked for a while, even her data sets from like 1986 there would be a sheet that went with it that had all kinds of stuff typed in. Like who went out there, the names of the people, like was it a cloudy day? I mean, everything, like maybe not all that. If it was like a whole summer, she wouldn't put every single day's weather, but like all the details are there. It's just good practice. So you have everything there. We call that metadata now, which is a fancy word for knowing what the data set variables are. You need a high quality or journal quality description of the methods somewhere. If you're writing a thesis, that'll be in your thesis. But even if you're just along the way and you don't know where you're going, early on, you should type out what happened. What are the methods? Potentially with pictures. For some reason, this is a baffling thing to me. For some reason, students don't appreciate, in general, the value of pictures. I don't get this. You take pictures all the time, right? I mean, you've got like you have a camera. This is so you must be into picture taking. Most students are. But what about picture taking of data stuff? Why aren't you doing that? That's weird. I can't understand it. So yeah, take pictures of what you're doing. I've got, and I'll show you some other time, probably. I mean, I would, every sample I collected back in the old days, I would take a picture of it. You know, so that you know what's there, and later if something comes up, you can go back and go like, okay, let me go back and look. It's sample number CV24 and see the picture of it. Here, oh, here's what happened. So you could, in your method section or in your folder, whatever, have pictures that explain what's happening. Okay. Don't do anything until you finish this. What will happen, and if you're on this, if you hear my voice, this is likely going to be the temptation for you. What's going to happen in reality is you're going to start your project. You're going to kind of not know what's going on. Your PI is going to be distracted, especially if it's a big lab or there's things happening at the university, whatever. Things are going to be like chaotic. You're going to get going in your project, and the timeline is going to press you. You're going to be like, I can't believe I'm running out of time. But it happens to every single student. Okay, like that's the norm. You run out of time. Time slips away from you. Impermanence. Look it up. Impermanence. It's a huge issue. And then you get towards the end of your project and you're just like, I just need to write this damn thesis so I can get out of here, man. The waves of Sarasota are calling. You can smell that bonfire from here. We're gonna get some White Claws. No laws with the claws, and you're heading to the beach. And like this whole friggin' fly thing is not so not going to be my problem anymore. So you get to the point where you're just like, I'm trying to get this thesis done, and you're liable to skip all this. You're going to want to. You're just be like, I'm not going to. Just going to get the data in as fast as I can and analyze it and just get this and make the graphs and share the graphs, and then graphs are done. Then go to my defense and get through the defense and then leave. I'm telling you, stop it. Don't do that. It's forbid. I forbid it. Whether you're here or not, I forbid it. It's bad practice. You got to be better than that. You took my class. You know that. You know better. Whether your PI knows better or not, you do. So stop. Make this happen. Okay? Make it happen. If you make this happen, you eliminate huge problems in science. So make the data, get the data ready early on. Don't do anything else. Don't start writing or anything else till you get this done. Now, data analysis, last point. Data analysis best practices. Here are a few tips. Number one, annotate your script regularly and with care. Again, you're going to be in a hurry. You're going to think, why do I need to know? I'm just going to do this. And you're busy. You're like doing one thing after another after another. So you're not going to want to do this. You got to trust me. You want to annotate your script so you know what's happening, because you will forget, unless some people in the class are much smarter than me. But for me, I forget what's going on quickly. like a few days pass, like if I'm working on an analysis and let's just say something happens, 2 weeks pass, it's very likely I'll forget that I ever did any of it. I won't even know. Like, oh, there's this big code I wrote. I don't even know. I mean, it's like that bad. I will forget what I was trying to do and what I was trying to get accomplished because I'm worried about whatever the next thing is. Okay. For most people, that's a real threat. You could lose a lot of time. So you need to just write down notes as you go. Here's an example. I just pulled this out of some notes that I had. This is before R. I used to just keep a notepad, like a TXT file, open all the time when I was doing analysis. And I would just like diary myself stuff so that I knew what was going on. So here's just like a note I pulled out just to share with you from March 13th, 2006. Here is your problem now. You set up a criteria that used 15% as a threshold for a fire year, but you did not know that FHX2 was thinking about recorder years, not the total number of samples. This is a problem because you followed convention and had null years prior to wounds and recorder years after, blah, 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 blah. What the hell? Anyway, there's some curse words that I've blotted out here so I don't offend your sensibilities. This is just an example. And I'll tell you right now, I have no idea what any of this is about. None, zero. That's not true. I know a little bit. But this is like important information that I could easily forget about 2 weeks from then. And then like have to redo all this. And I might not even be as sharp as I was in this moment because it looks like in this moment, one thing I read from this is like, I was on that. I was 100% on that. Like I mentally engaged. So you need to leverage that by taking lots of notes in your script. And you'll see it in the class script I share. I have hashtags and lots of notes. Just like get yourself in the habit of taking lots of notes. And the best practice would be take notes in such a way that if a stranger read them, they would know what you're doing. Because a stranger might be the person, or at least your PI or some other grad student in the future, might be the person who needs to figure out what's going on. Number 2, share your results with your PI and collaborators early and often. Here's again, every single one of these points, I could just say is of incredible importance to your success and your empowerment. A dreadful mistake that students make, graduate students especially, or undergraduates who are doing a thesis. You get started in a project, you're trying to get to the end, and at some point you start to do analysis, anxiety starts to ramp up. You start to feel like, I wonder if I'm doing it wrong. This doesn't seem like what I thought. These results don't match what my professor was saying. This doesn't seem to be the right thing. These flies, maybe they got infected by a fungus, or there's something's wrong. This is not good. You might not even be right about any of that, but it's just like some people, not everyone, you can start to develop these kind of pathological cycles in your mind. You're just like, is it right? Did I do it wrong? And then because of that, and it's common enough that I think it's worth having a point on here. Because of this, you don't share your results. You tend to hoard them. And then by hoarding the results, you don't give your PI and or collaborators the opportunity to help you. To be like, oh, because look, I've been doing this since the first data analysis I did that was a serious data analysis was in 1998. Were you born then? How many people, raise your hand. Were you born in 1998? I don't even know. Alexa, what is 2022 minus 1998?
2022 minus 1,998 is 24.
24. So few people on this call were born when I was doing data analysis for the first time. All I'm saying that for is just to say, I've got pretty good instincts about what's a problem and what's not. But if you don't show me the data, I can't tell you like, oh, hey, here's the thing. So I want you to get in the habit of, I said this in a previous one, like literally print the results out, send it. You can send an email, it's fine. With like a PDF just of the figures. And just like, look at these, please. Or have a meeting where you're like, or print them. Here's a great idea. Print it out, just the figures, and staple it like this. I've got some actually right here. And just like put that in your PI's mailbox or hand it to them in the home. Best yet, set a meeting and literally put them on the desk. And do it almost as soon as you possibly can. Earlier is better because it increases the probability that the person is going to be able to identify nice tracks for you to go down, identify problems. Hey, that's a bad one. No. Leave that alone. Focus over here. And it could change the whole track of your thesis, your whole trajectory. And you can't wait for your PI to ask you for the data. They're just like, they're lost. Okay. Just lost. Forget it. It's just like, if you wait for that, again, this is like the key problem between undergraduate and graduate school. Undergraduates get in the mindset of like due dates. I've got to meet this due date. Graduate students, there might not be any really, none. You're just in the ocean. You have to figure out how you're going to generate energy and momentum for your project, and you have to own that and take responsibility for it. And if you don't, you will fail. So you can't wait. Like I'm going to wait around for my PI to ask me for the figures. What? No. Give them the figures as soon as you make them, and start the conversation. This is drifting. This next bullet item is drifting into the area of like how to write a scientific paper. But I just want to give you this tip. And it's a key problem that a key error students make, graduate students, especially if you're just getting started. Once you've got a set of figures, like, oh, I've got all these figures. I've got these graphs. Oh, here's these graphs that are going to be in my thesis. It's going to be my peer-reviewed journal article. I've got these 5 figures. Be sure that when you're writing, that your methods match the figures. Because here's the problem. You started your study and you wrote a bunch of stuff up and you did all these things. But some of those things got dropped. And some of the stuff doesn't match the figures. So you need to start thinking about like welding together how you're writing with what you did in the end, ultimately this. And like when I read, if you give me this figure like that, like this is going to be in my thesis. As a reader of this, when I go back to your method section, I need to know what this is about. You need to just literally, again, old-fashioned, right? Here's my recommendation. If you got a graph of your honors thesis data or your master's thesis data or whatever, put it on a sheet by itself like that and print it out and hold it in your hand and look at it. It's like, and think about it by itself. You're just thinking about this figure. And then on your computer screen, when you're typing your results or typing up your methods, keep looking at this. Does what you're writing make sense in relationship to this? It's a big mistake. And it makes science writing much worse, and it makes your thesis process much slower, and it causes all kinds of hiccups and issues with reviewers, et cetera, if what you're writing does not match what you've presented in the figures and the tables and the analysis. Okay. Here's a simple test from like 1 through 3. and you can just ask yourself this before you graduate. In less than 2 hours, can a reasonable expert, not someone like someone who's reasonably good at R, can they recreate the figures from my thesis from the stuff I've got stored? Can somebody, like if I just picked a stranger out, I went to a conference, I'm like, okay, come here, stranger. Sit down at this laptop. Here are my figures. And here's the stuff I've got stored on this folder. CSV's code method section written out. Remember that stuff? See if you can recreate these figures. If you're like, there's no chance that could possibly happen, then you've made a mistake. If the answer is no, then make that be the case. It means that the final CSVs are available and curated, the scripts are available and curated, and the annotation and metadata are available. It's like minimum question to ask yourself. Item 4, publish the data set. Once you've gotten to this point, back to Chris Lorty's thing, he's saying, publish the data set, publish the data descriptor. Why are you doing this alone in secret? Doesn't make sense. Doesn't make sense. Okay, how do you do that? Well, there's lots of places where you can publish data sets. You can post it to the thing called, this is a NOAA thing. There's a thing called the International Tree Ring Data Bank. And I actually, in one of my papers years ago, used a reference chronology. This reference chronology right here, see this one? Those are data from that International Tree Ring Data Bank. Somebody else can use it in the future. And I cited the person. It's great. This is vegetation data, Veg Bank. Here is the Knowledge Biodiversity Network. This is for eco-informatics data. This is the International Nucleotide Sequence Database Collaboration. There's data source, there's ways to share data all over the internet. This is the Arctic Data Center from the National Science Foundation. There's lots of ways. I'll also say, the way I share all the data from my lab is a thing called eCommons at the University of Dayton Library, the environmental data archive from my lab, McEwen Lab. All of our data are available. Well, I mean, we're still working on some, but things that have been through the process get posted on there under this eCommons, and it's available for anyone to access. And then here's the tip. You publish the data set, even on an archive like this. And you don't do that unless it's very clean and ready to go. You've done the exploratory data analysis. You don't have negative values for fly eye diameter or sperm tail, whatever. Something you're measuring. And then you go, okay, it's all ready to go. Post it on there to eCommons. And then you can cite the data in the paper. This is an example. So this is a sentence from a manuscript we published. We compared our methodology against 2 methods for blah, 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 and then cite Maloney et al. 2018. This is a citation, right? And then in our literature cited, it's Maloney, Borth, Chapman, and McEwen 2018. That's Maloney et al. 2018. But this is a, see the brackets, data set. It's not a manuscript, it's a data set. And then we've got this doi.org. If you clicked on that, which we can't now, obviously, it goes to that archive. And it's a CSV file. So you can literally go get the data yourself. And in fact, the ultimate transparency is something like this. And maybe I'll send this to you in it, or you can just, yeah. I could literally just copy this and email it to you. And you could literally copy it out of the email and paste it into R and hit go. And the figure from the manuscript will appear. Because this is the CSV file. See it? Posted on eCommons. So this code, this is our code saying, hey, type is this read.csv file, or it's saying type equals read.csv, this file, which is posted on that eCommons site. And then all the rest of the code points back to this. See type right here. This is some code you don't know about, subsetting, whatever. Like, oh, I made a figure. So you can literally publish your whole script as a supplemental document. And then a reader could literally go to R, copy it, paste it, bam, and have everything you did. Now, publishing scripts is something that's not that common right now, something to do, work towards, but I'm definitely doing this. We publish the data set before we ever publish the paper, and we cite the data set in the paper. See that? Also, note, Maloney gets another citation because of the data set. It's great. And not all data that we post to eCommons gets cited, because not all studies result in a peer-reviewed journal article. On the other hand, you can go back, if somebody else wanted to, could go back and be like, oh, you know what, we're really interested in stream chemistry data from these sites. In fact, story number 6 or something, we do have data from a stream where there was going to be this airport thing with all wood, and here nearby, they were going to mess up this prairie. And there was a moment where it looked like our stream data that we collected from this in this study might be useful for somebody with a lawsuit or whatever, trying to stop this development. It's already posted on eCommons. You can have it. It's no problem. It's there for anyone to access. It didn't work out that way, but it was possible. Last point. As you're about to graduate, I've already said you should create a folder in which you contain everything. But one thing to do towards the end is to make sure, okay, I've got a visitor here. See that? Little guy. Hemiptera. Any hemipteran experts in the room? We used to have a hemipteran expert in the room. Transfer your data products, your scripts, and all of the results before you graduate. So before you're at your defense, last check. Have you given everything into a folder that your PI, collaborators, other students, lab, techs or whatever have access to? Everything is there. Then, once you've done that, you can go to the bonfire in Sarasota and be like, it's cool, man. You got everything. Now, your PI might still want you to do the analysis, but you just be like, dude, no, I got a date. I'm going to skip some rocks in the moonlight. All right, that's it for this lecture. Hopefully that was useful. Hopefully you'll take it like super seriously and be like, dude, man, I'm going to do all those things, because those are good tips for you to become empowered and a better scientist. So we'll see you in the Coding Club.